welcome to the fourth video for Life on Earth. You should now know all about abiotic factors, and you should be able to pause the video and answer the starter question before I go over the answer. So the question says, for light intensity and soil moisture, identify a possible source of error for a named measurement technique and explain how it might be minimized. So you need to put your measurement technique, your source of error, and how to minimize it. So the answers to this would be, for light intensity, the measurement technique would be by using a light intensity meter to measure light intensity. Your source of error would be casting a shadow over the light intensity meter sensor, and you would minimize this by avoiding standing over the light intensity meter and avoiding casting a shadow over the light meter sensor. Your other measurement technique, because you needed to measure soil moisture, would be to measure soil moisture by using a soil moisture meter. Your possible source of error would be that the probe is contaminated with damp from the previous soil sample, or that your probe is inserted at different depths. So to minimize this, you would need to wipe your probe in between each measurement and make sure it's obviously wiped and dried. So if you got that all correct, well done. If you struggled to get the answers, then remember you can watch the last video or parts of the last video that you're struggling with as many times as you want. So today we're going to be learning about the third key area, which is photosynthesis. So if you want the worksheets to go with the video, remember to pause the video and go to Free Life on Earth, Free Photosynthesis, 2 consumables and activities. And then the PowerPoint is found in PowerPoints and is called Free Photosynthesis. So today we're going to be learning about photosynthesis. Our learning intention is to give the word equation for photosynthesis, to describe the two-stage process of photosynthesis, to describe the light reactions, to describe the carbon fixation reactions, and to identify the possible uses for the sugar that has been made. To be successful, you will be able to state that photosynthesis is a two-stage process. You'll be able to describe the light reactions as being where the light energy from the sun is trapped by chlorophyll in the chloroplast and is converted into chemical energy, which is used to generate ATP. Water is split to produce hydrogen and oxygen. Oxygen diffuses from the cell. Then you will also be able to describe carbon fixation as a series of enzyme-controlled reactions which use hydrogen and ATP produced by the light reactions with carbon dioxide to produce sugar. Then finally you'll be able to state that the chemical energy in sugar is available for respiration or for the sugar so it can be converted into other substances such as starch for storage and cellulose for structure. Can you remember what the words consumer and producer mean? Pause the video and try to write out the answers. So the answer is that a consumer is an organism that gets its energy by feeding on another organism. It can't create its own energy. Whereas a producer is something that produces its own food, and they are usually plants. But how do they produce their own food? Pause the video and write down how you think that producers produce their own food. If you thought that plants produce their own food by photosynthesis, well done. Now we need to look at what photosynthesis is, where does it occur, how does it occur, what are the substrates and products, and why does it happen. We already looked at the structure of the leaf in Unit 2 of Plant Transport. Pause the video and try to label all of the parts of the leaf before I go over the answers. So the answers is, the first part is the waxy cuticle, then you've got the upper epidermis, then the leaf vein, which is made up of your xylem and your phloem. Then you've got your palisade mesophyll layer, you've got your spongy mesophyll layer, you've got an air space in your spongy mesophyll layer, you've got your stomata, you've got your guard cells. So obviously your stomata is your pore, and then you would also have your lower epidermis, which would be your. Well done if you managed to get that correct. If not, then you might want to look back over the video on plant transport. Also, this table is just revision, so you should be able to fill in the leaf part and the function for all the tables. So pause the video and try to do that before I go over the answers. So the waterproof outer layer will be your waxy cuticle. Your protective outer layer, which is transparent, will be your epidermis. So your upper one obviously is at the top and your lower one will be at the bottom of the leaf. What transports water in a leaf will be your xylem vessel, and what transports glucose sugar will be your phloem. Now remember xylem because xylem begins with X and it's near W in the alphabet. Phloem starts with PH, so it sounds like F, so it sounds like food, which would be your glucose, and sugar for plant. Now, the two parts that you need to know about the leaf for photosynthesis is the palisade mesophyll layer, and this is because you know that it contains lots of chloroplasts. So it is the primary region of photosynthesis. So this is where photosynthesis occurs. So photosynthesis occurs in the leaf, and it occurs in the palisade mesophyll layer. The other layer that you need to know for photosynthesis is the spongy mesophyll layer. Now, the spongy mesophyll layer is loosely arranged for gas exchange, and that's why it's like sort of circular in shape, and it is the secondary region of photosynthesis, so it also contains some chloroplasts. Now, your moist airspace is where it allows gases to dissolve, so obviously your oxygen and your carbon dioxide. And then your stoma or your stomata allow your carbon dioxide in for photosynthesis, and they allow your oxygen to leave the leaf. And then finally, your thing that opens and closes the stomata is the guard cell. So well done if you managed to get that correct. If not, then you need to go back and watch plant transport. We also looked at the stomata in plant transport. So stomata are the small pores on the bottom of the leaf. So there's some on the top, but most of them are found on the bottom of the leaf. They are open during the day for gas exchange, so they allow carbon dioxide in and oxygen out. So we need carbon dioxide for photosynthesis, so that is why they're important. So this allows the first stage, which is called the light reactions in photosynthesis, to take place. The stomata close at night, which reduces water loss, as the light reactions cannot take place at night, because obviously it's dark. And the guard cells control the stomatal opening. So remember that they open and close by osmosis, so the movement of water from a high concentration to a low concentration. So they open when they gain water and become turgid, and they close when they lose water. So well done if you know all that. If you don't, then you might want to watch plant transport again and watch the start of this video again. So what actually is photosynthesis, and how do plants make their own food? You should know the equation for photosynthesis from previous years, like third year, second year, and first year. So we learn how to remember respiration by the phrase "go cow." So photosynthesis is the opposite, so you can remember it by the phrase "cow go." So the substrates or raw materials that you need to start photosynthesis are carbon dioxide and water. Then it needs to be in the presence of light energy, which you get from the sun, and it's absorbed by chlorophyll. Then your carbon dioxide and your water react to form and make glucose, which can be used as a food product, and oxygen, which is used as a byproduct. Now, a byproduct is just another way of saying waste product. So your photosynthesis can then change this light energy from the sun into chemical energy in the form of ATP. And we've heard of the word ATP before. So make sure you know this equation, make sure you can write all of this down before you move on to the next part of the video. So we know that photosynthesis occurs in the leaf, and it primarily occurs in the palisade mesophyll layer, and it also occurs in the spongy mesophyll layer. But what organelle does it actually occur in? So pause the video and try to write down exactly what organelle it occurs in before I go over the answer. So you should know already that chloroplasts are the site of photosynthesis, so chloroplasts would be the organelle that it occurs in. So light energy is trapped in the green pigment called chlorophyll. Chlorophyll is found in a discus-shaped structure called chloroplast. So make sure you know this before you go to the next part of the video. What controls photosynthesis? Pause the video and write down what you think controls photosynthesis. So photosynthesis consists of a series of enzyme-controlled reactions. So enzymes control photosynthesis. So if you plot photosynthesis against temperature, you would end up with the same curve as if you plotted enzymes against temperature. So try to draw the enzyme curve that you know already in your jar and pause the video. So the answer is that it should look like this, and this is because the rate of photosynthesis increases as the temperature increases, as your enzyme collisions will increase until it reaches its optimum. So its optimum of an enzyme is usually about 37 degrees. And then above the optimum of the enzyme, the rate of photosynthesis and enzyme activity would decrease because enzymes denature at high temperatures, so the active site changes shape and it can no longer complementary fit with the substrate. So this is a usual exam question, so you usually get asked about how the temperature is affecting photosynthesis, and it's usually a 2-marker question. So for your first mark in an exam question about how temperature controls photosynthesis, you would need to say that photosynthesis is controlled by enzymes. So first mark, photosynthesis is controlled by enzymes. Your second mark is for saying how the enzymes are affected. So for example, the enzymes increase the photosynthesis activity until they reach their optimum. Above their optimum, the enzymes denature. So you'd obviously have to tailor that to whatever that you were being asked about. You need to know the 2 stages of photosynthesis and what their names are. So photosynthesis is a 2-stage process. The first stage is called the light reactions and it consists of several light-dependent reactions. The second stage is called carbon fixation and it is temperature dependent. So make sure you know this before you move on to the next part of the video. Let's look at each stage in more detail. We know that the first stage is called the light reactions. In the light reactions, the light energy from the sun is trapped by chlorophyll in the chloroplast and is converted to chemical energy, which is used to generate ATP. The light energy is used to split water to produce hydrogen and oxygen. Remember, water is H2O. The hydrogen is passed to stage 2, carbon fixation, whereas oxygen diffuses from the cell as a byproduct. So you need to learn all of this before you move on to the next part of the video, and it might be useful to write down the diagram as well. So you've got your light energy, it creates ATP, so ADP add PI gives you ATP, and that needs to be taken to the next stage for carbon fixation, so stage 2. Your light energy also splits your H2O, which is water, into hydrogen. Hydrogen is taken to the next stage for carbon fixation. And it is also split into oxygen, and your oxygen diffuses into the air. It's a byproduct. So remember the word oxygen, byproduct. Right, so let's look at the next stage. The second stage we know is called carbon fixation. So in the second stage, carbon fixation is a series of enzyme-controlled reactions which use hydrogen and ATP produced by the first stage, the light reactions, with carbon dioxide from the air to produce sugar, which is also called glucose. So you've got your hydrogen, which you got from the first stage, you've got your ATP, and both of which are combined with carbon dioxide from the air to form sugar, which is called glucose. So you need to make sure you know this before you move on to the next part of the video. So carbon fixation, a series of enzymes controlled reactions which uses hydrogen and ATP with carbon dioxide from the air to produce glucose. So in summary, stage 1 is called the light reactions. The light energy is trapped by chlorophyll in the chloroplast and it's used to split water. The water is split into oxygen, which is used as a byproduct, and hydrogen, which is moved on to stage 2. And it is also used to generate energy. So it is used to make ATP from combining ADP and PI. So then our ATP and our hydrogen are passed on to carbon fixation. So in stage 2, which is called carbon fixation, our carbon dioxide from the air and our hydrogen with ATP are combined together to produce glucose. So obviously first stage, light reactions, second stage is called carbon fixation. So write this out over and over again and make sure you can say both the stages off by heart without looking at it before you continue through the rest of this video. So we obviously know that our end product is glucose, and that's what's made from photosynthesis. But what can our glucose then be used for? So the chemical energy in sugar is available for respiration. It also can be converted into other substances. So it can be converted and stored as starch, or it can be changed into cellulose and used as structure. So if you remember, the cell wall in a plant is made up of cellulose. So make sure you know the three things that glucose can be used for. So respiration, storage for starch, and also structural, and that would be in the case of cellulose. So make sure you know that before you move on. So now try to write down the three things that I said glucose could be used for. Pause the video and try to answer it before I go over the answers. So the answers are that glucose can be used for respiration, so it can be broken down during respiration to provide energy for the cells. It can be used for starch, so it can be converted to starch as a storage carbohydrate, or it can be converted to cellulose. So converted to cellulose, which is a structural carbohydrate, which is the main component of the cell walls. So remember to write this down and remember it before we move on. Well done if you managed to get all of them correct. Let's review your knowledge so far. So pause the video and try to answer these questions. So the questions are: name the raw materials for photosynthesis, name the pigment used to trap light, give the word equation for photosynthesis, name the two stages of photosynthesis. So the answers are that water and carbon dioxide are the raw materials for photosynthesis. Chlorophyll is the pigment used to trap light. The word equation for photosynthesis is water and carbon dioxide reacts to form, which is the arrow, glucose and oxygen. And the stages of photosynthesis are stage one is the light reactions and stage two is carbon fixation. Well done if you got that correct. If not, you need to go watch the video again. Pause the video and try to answer these harder questions. So the harder questions are: name the form of energy light energy is converted to, describe the three uses of glucose in a plant, what happens to the oxygen produced, and name the products of the light reaction that are used in carbon fixation. The answers are that light energy is converted to chemical energy in the form of ATP. Glucose can be stored as starch, it can be used for energy and respiration, or it can be used as a building material when used as cellulose, so structural. Oxygen is passed to the atmosphere through the stomata and is used like it is a byproduct. And then for 4, hydrogen and ATP produced or gained in the light reactions are used in carbon fixation. So again, well done if you got that correct. If not, then go look at this video again. So let's try some SQA questions. So pause the video and try to answer the questions before I go over it. So the question says, the diagram below shows two stages of synthesis, and then it's got the diagram, and then it says, name the pigment in chloroplast which traps the light energy required for photosynthesis. For B, it says, in the light-dependent stages of photosynthesis, light energy is converted to chemical energy. Identify substance A in which the chemical energy is stored. For C, it says, identify substance B produced in stage 1, which is required for stage 2. Give one substance into which the sugar produced in stage 2 can be converted, and then explain why temperature can limit the rate of photosynthesis. The answer for A would be chlorophyll. So chlorophyll is what traps the light energy. And in identify substance A, so let's look at substance A. So we've got ADP and PI forming substance A, so we should know that that should be ATP. And then identify substance B. So let's look for substance B. So we can see that water is being split into the byproduct, so that must be oxygen there. And then it also gets split into hydrogen, so hydrogen would be our answer. And then give one substance into which the sugar produced in stage 2 can be converted, so we should have got starch or cellulose. And then explain why temperature can limit the rate of photosynthesis. So we should have put that photosynthesis is controlled by enzymes. So if the temperature decreases, it slows enzyme activity. So well done if you managed to get that all correct. If you didn't, then go watch the video again. So another SQA question is, the diagram below shows two stages of photosynthesis, and then you've got your diagram. It says name stage 1. Identify the byproduct released during stage 1. Name products X and Y produced in stage 1 which are required in carbon fixation stage, and describe the role of carbon dioxide in the carbon fixation stage. So make sure you try to answer the questions before I go over the answers. So the answer would be that stage 1, we can see the light energy is getting used to do all these things, so it's going to be called the light reactions. Identify the byproduct released during stage 1. Well, we know that water is split into hydrogen and it's split into oxygen, which is the byproduct, so it would be oxygen. Then it says name products X and Y produced in stage 1 which are required in carbon fixation. So we know that we get water split into hydrogen and oxygen, and it would be hydrogen in this case because it's the only thing that moves to carbon fixation. And Y, we've got ADP and PI, and we know that produces ATP. Then we get asked to describe the role of carbon dioxide in the carbon fixation state. So we should have put combines with hydrogen using ATP to produce glucose, or you could have put instead of glucose sugar. So well done if you managed to get that correct. If not, then go back and watch the video again. You should have all tested the leaf for starch in class. You might have done it in first year, you might have done it in second year or first year. So we know that if the leaf has starch, then the leaf must be photosynthesizing because it's obviously a buildup of glucose. And we know that we test for starch using the chemical iodine. So iodine is a brown chemical and it turns blue-black if starch is present, and then if starch is not present, then it'll just stay brown. So to do the experiment, you first need to boil a leaf in boiling water in a beaker, as it breaks down the cell wall of the leaf and softens it by bursting the cell membrane. So that's the first stage. Then for the second stage, you need to put the Bunsen burner off. It is important that you put the Bunsen burner off for this point because ethanol is highly flammable. Then you would put the leaf into a test tube of ethanol, which you place into the beaker. So the ethanol will then remove the chlorophyll and your leaf will turn a pale, like white, sort of transparent colour. Then you'd add the leaf to the dimple tray after washing the ethanol off of it, and then you would add iodine to the leaf. Now if the iodine obviously stays brown, it means there's no starch. If it turns blue-black, then it means the starch is present. So sometimes the SQA will ask you a question about different plants or different leaves of a plant set up in different conditions, and it asks you which plant or leaf can photosynthesize. So try to work this question out for yourself. So which of these leaves can photosynthesize? Is it A, B, C, or D? Before I go over the answer. So the answer would be that the leaf that can photosynthesize is D, and that's because it has access to light. It's got access to carbon dioxide, nothing's blocking the carbon dioxide, so it'll be able to carry out the stages of photosynthesis. It can't be A and it can't be B because they're both in plastic bags and they've both got a carbon dioxide absorber chemical. So that means that they're not going to have carbon dioxide, so they won't be able to do the second stage of photosynthesis. It also can't be C because it's in an envelope which is blocking the light, so it will not be able to do the light reactions. So the only leaf that can photosynthesize would be your D. So well done if you managed to get that correct. Now chlorophyll is really important, and you can tell this because only the green regions react positively with iodine, which means that they turn blue-black, so that shows that starch is present. Whereas the white parts of the leaf, they do not turn blue-black, so they do not react positively. Therefore, it is concluded that chlorophyll is only contained in the green part and is necessary for starch production and photosynthesis. So only your green part of your plant will photosynthesize because it contains chloroplasts and chlorophyll. The white part that doesn't contain chloroplasts and chlorophyll will not photosynthesize. Now try this SQA question by pausing the video and coming up with the answer before I go over it. So it says the diagram below shows an investigation into factors affecting photosynthesis in a green plant. And then you've got your diagram, and then it says which of the following statements is true. So A, leaves X, Y, and Z produce sugar. B, only leaf X and Y produce sugar. C, only leaf X produces sugar. And D, only leaf Z produces sugar. So the answer would be, let's have a look. Well, let's look at Y. So why is it going to produce sugar? No, because it's not got access to light. Then let's look at X. So it's in a plastic bag and it's also got carbon dioxide absorbing powder, so it's not going to have carbon dioxide, so it's not going to produce sugar. So the only one that's going to produce sugar is Z, so it should be D. So well done if you managed to get that correct. If not, then you need to go back and watch the video again. We have now come to the end of the lesson, so you should now be able to describe the light reactions, which is when light energy from the sun is trapped by chlorophyll in the chloroplast and is converted into chemical energy, which is used to generate ATP. Water is split to produce hydrogen and oxygen. Oxygen diffuses from the cell. You should also be able to describe carbon fixation as a series of controlled enzyme reactions which use hydrogen and ATP produced in the light reactions with carbon dioxide to produce sugar, and you should be able to state that chemical energy in sugar is available for respiration, or the sugar can be converted into other substances such as starch for storage and cellulose for structure. Well done if you can confidently do all of that. If you can't, then you need to go watch this video again. That is the end of the video tutorial. Well done. Thank you for listening.